Hello everyone, welcome back, and in today's class we will learn about female reproductive part of flower. So what exactly is female reproductive part in flower? It is gynoecium that we learn in the previous class as well. So gynoecium is what? It is the female reproductive part in the flower. Now gynoecium can have multiple units and these individual units are known as pistil. So pistil is again a female reproductive part in flower. Then what is the difference between gynoecium and pistil? So let's say if there is only one pistil, we call it pistil only. If there are more than one pistil in a flower, collectively we call all the pistil what? Gynoecium only. Clear? So this is the difference between gynoecium and pistil. Okay. Now gynoecium can have a single pistil or more than one pistil. Yes. If the flower contain only one pistil or one carpel. Pistil and carpel both are same. If the flower contain only one pistil, we call it monocarpellary. If the flower contain more than one pistil, we call it multicarpellary. Clear? Now think about it. If the flower contain more than one pistil, then these pistil can be fused together or can be free. Now what is the meaning of this? Let's say there are 3 pistil in this flower. If there are 3 pistil in this flower and if they are free from each other, then what we call it? Then we call it apocarpus. Why? Because the pistils are free. Now just imagine another situation. If all these pistils are not free but they are fused together with each other. They are not free but they are fused together with each other. If they are fused together with each other, in this situation, this type of carpel is known as syncarpel. So clear? Let's revise it one more time. Pistil is what? It is the female reproductive part in flower. If only one pistil is present, we call it monocarpellary. If more than one pistil is present, that means it is multicarpellary. Now, in case of multicarpellary also, if all the pistils are fused together, in that case we call it syncarpus. And if the pistils are not fused together, they are not fused, they are free. So this type of pistils are apocarpus. Clear? So this is just an example of what? Fused pistil and free pistil. Yes. So if the pistil, so look at the top, all the pistils are fused together. If the pistils are fused together, what we call it? We call it syncarpus. Syncarpus. If the pistils are free from each other, if they are free, then this situation is known as apocarpus. Clear? Okay. Now let's come to the structure of each individual pistil. Yes. So pistil again consists of 3 parts. There are 3 parts in an individual pistil. These parts are stigma, style and ovary. So which part is stigma, which part is style, which part is ovary, that I will explain you. So stigma is again, I can say, the uppermost part most of the time. Now see the word stigma. This place is where the pollen grain will come and attach or stick. So this part is stigma. Now below the stigma, what is present? A long cylinder kind of part. So we call it style. So style is something which connects stigma to the ovary. Now the bottom part is what? Ovary, right? So 3 parts that we just learned. The topmost part is known as stigma. Below the stigma, what we have? Below the stigma, we have style. Yes, style. And style connects stigma to the ovary. So bottom part is ovary. Clear? Now, what is there inside the ovary? Ovary contains what? Inside ovary there is ovules. Okay, and this ovule help in what process? Formation of female gametophyte. Clear? Now let's come to the structure of ovule. So for this, first we should clearly know that ovule is present inside the ovary. Ovary is a part of pistil. Pistil contains three parts. There are three parts of pistil. These are stigma, style and ovary. A stigma is the place where pollen grain come and stick or attach, and ultimately with the help of style these pollen grain will reach to ovary, and inside ovary what is present? Ovule. Inside ovule they will form what? Embryo after fertilization. Now what we will do, we will deep dive inside the structure of ovary. Inside the ovary what is present? Ovule. Ovule is present inside ovary. Now we are talking about female reproductive part, right? So here the word is megasporangium. For male reproductive part, the word was, for male spore, the word was microsporangium. Clear? This you need to remember, microsporangium and megasporangium. So now this part contain the structure of ovule, very important. There are a lot of small words that you need to remember. So these words are ovule, funicle, hilum, integument, micropyle, chelygel and nucellus, and ultimately embryo sac, that we will learn one by one. So first we'll try to learn the structure of ovule with the help of a diagram. Now think about it. What it is? It's, let's say this structure is present inside ovary. The complete structure is present inside the ovary. Now imagine, if this structure is present inside the ovary, or the ovule is present inside the ovary, there must be something through which the ovule is attached to the ovary, right? So what is this particular thing? Placenta, obviously. So with the help of placenta, ovule is connected to the ovary. Clear? Now the second thing, very important, there is one more structure between placenta and what? And ovary. What we call it? Funicle. So in simple word, what we can say, ovule is attached to the placenta by a stalk or a structure. We call it funicle. What we call it? We call it funicle. So funicle is something, very important word. Funicle is something which attached or which connect placenta and ovule. Yes, clear? Can I say funicle is a part of ovule, because placenta a different structure? Now we are learning about ovule. So does funicle is a part of ovule or not? So again I'll repeat the same sentence. Ovule is attached to the ovary by a stalk called funicle. So yes, it's there. So funicle, you must remember, it is that part of ovule which directly connected to placenta. Now one more important word, these two words are important, rest are very simple. Another word is hilum. Now what is this hilum? Hilum is nothing but it is a junction between ovule and what? Funicle. Clear? So between ovule and funicle what is present? Hilum. Between ovule and funicle what is present? Hilum. Clear? Now think about it. These two words are clear or not? Funicle and ovule. We will revise it one more time. Funicle is a part where the ovule got attached to placenta, and hilum between funicle and ovule. So funicle and hilum is done. Next let's come to other part of the ovule. What are the other part of ovule? Now ovule itself is covered by some kind of covering or layer. So these protective covering in the ovule is known as integument. These covering are known as integument. Clear? These are what? These are integument. Very, very important. So there are two integument, outer layer, outer integument, inner layer, inner integument. But we can observe one more layer around the embryo. So what is this layer? This is nothing but nucellus. Now what is a nucellus? It is the part inside the ovule. It is also a part. What's the purpose of this part? It stores some food. Okay. So it's the function of providing nutrition goes to nucellus. So you can remember easily nucellus, nutrition, nutrition, nucellus. What are these integument? These are the covering for the protective purpose, right? So till now we covered about funicle. What is funicle? Funicle is a stalk that hold ovule in place. So how come? With the help of placenta. Clear? Now hilum is a junction between funicle and ovule. Clear? So think about it. This part is hilum. This is what? This is hilum. And this uppermost part, we can say it will go a bit up. So this is funicle. And these two covering are nothing but outer integument and inner integument. But these two covering are not continuous. There is some space at the top. These are not continuous, but there is some space at the top. Any idea why there is some space at the top? Think about it. Let's say if there is no space, how the male gamete of, how the sperm, or we can say the pollen grain or the male gametophyte will enter inside the ovule. There must be some opening, right? So with the help of this opening, the pollen, the content of the pollen tube will ultimately released. Okay, so this opening, there is this small opening, we call it micropyle. Very important. This opening is known as micropyle. Very important. Now this opening is present at the top. This opening is known as micropyle. So this top end of the ovule is known as what? Micropylar pole. There are two poles. The upper pole where micropyle is present is known as micropylar end. Clear? This upper end where micropyle is present is known as micropylar pole or micropylar end. Now there is another end as well, right, which is it opposite, very far. So this end is known as, not very far, but yes, compared to micropylar pole it is far. So this one is chalaja end, chalaja end. Okay, so it's far, opposite, chalaja end, chalaja end. So chalaja pole. Clear? So this is all about structure of ovule. Very important. Let's revise it one more time. Structure of ovule. So what is ovule? First of all, it is structure which is present inside the ovary where seeds develop, okay, in future after fertilization. Now without fertilization, in some plant it can develop, but we are talking about a situation where pollination is required. So ovule is a structure present inside the ovary. Now this ovule is attached to placenta. How the ovule will attach to placenta? With this structure we call it funicle. Funicle. Okay. Now there is a junction between ovule and funicle. So again, let's say this is the ovary and this ovule. This ovule is attached to placenta. Okay. So how this ovule got attached to placenta? With the help of what? With the help of this structure which we call funicle. Now there is a junction between funicle and ovule. What we call this junction? This junction is nothing but this junction is known as hilum. This junction is hilum. Very important. These two words you need to remember. funicle and hilum. Integuments, protective layers surrounding the ovule, and these protective layers are not, we can say, continuous. There is a gap, there is an opening, we call it micropyle, which is present at the top. So the end where micropyle is present, that part is known as micropylar pole, and opposite of it is known as chalazial pole. So basal part, which is at the base, is known as chalazial pole, and at the top part where micropyle is present is micropyle. Clear? Now inside these covering, what is present? Inside these covering we have reserve food material as well. So these reserve food material is known as, or the layer where reserve food material is present is known as nucellus. Clear? So that's all about the structure of ovule. So these are 7 words, very important. You have to remember all these 7 words. Now, so till now we covered what is female reproductive part in flower, what is pistil, different part of pistil, stigma, style and ovary, what is present inside ovary, it is ovule, what are the different part present inside the ovule, that also we already covered. Now we'll talk about megasporogenesis. Now what is this megasporogenesis? Now we are diving deep inside the ovule. So megasporogenesis is nothing. It is the process of formation of what? Process of formation of female gametophyte ultimately. Very important process. So how this process will start inside the ovule, that we need to cover. So think about a situation. So for example, let's say as the ovule develop, when the ovule develop, a single cell from the nucellus itself. So for example, let's say there are many cells present in the nucellus, right? Integument, the outer integument, inner integument, but some cell in the nucellus, which the number is very less, so one cell will decide to become a megaspore mother cell. So think about it, there is this cell inside the nucellus which function as megaspore mother cell. Now what this megaspore mother cell will do, it will undergo in the process, in the process of meiosis, meaning what? It will divide mitotically. Mitotically? No. Haploid, in a haploid way. So for example, there is this cell, we call it megaspore mother cell. This cell undergoes meiosis division. Okay, meiosis means haploid division. So from single cell, 4 cell will produce, and all of them are haploid. Let's say megaspore mother cell is diploid. These 4 cells are not diploid, they are haploid. Clear? Now there are 4 cells now. So out of these 4 cells, 3 cells degenerates, and only 1 cell will remain functional. Clear? So 3 cells will go in the process of degeneration, and only 1 cell will become active. Clear? And this 1 cell will ultimately work as megaspore or function as a megaspore and develops ultimately into female gametophyte. Clear? Very simple, right? Now let's say this is just a diagrammatic representation of megaspore mother cell and how more than 1 cell will form from a single megaspore mother cell. So nucellus, we already know this is a nutritional we can say content reserve in the ovule. This top end is micropylar end, right? and let's say the cell present here, it is megaspore. This is megaspore mother cell. So megaspore mother cell will divide first and form 2 cells, and ultimately it will form 4 cells. Right. So from one single cell 4 cells will form. Clear? And this is known as megaspore tetrad. 1, 2, 3, 4 cells are formed. So out of these 4 cells, only 1 cell will remain functional, and 3 cells, what happened to 3 cells? They will degenerate. Okay. Now think about this one single functional megaspore, that is already haploid. Yes. So this single functional megaspore will again divide mitotically to form 2 cells. So N, N and N. This is mitotic division, and these 2 cells will again form what? 4 cells. Okay, these 2 cells will again form how many cells? 4 cells. So 1, 2, 3 and 4. So total 4 cells will form by 2 division of what? Functional megaspore. Clear? Now what will happen next? Now so we know inside the megaspore, or we can say inside the ovule, there are 8 nucleated structure present, right? Now see, there is this word free nuclear division, meaning what? The nuclear division happened before the formation of cell wall, and formation of embryo sac. After 8 nucleated stage, cell wall form and organize into female. So it's not like suddenly the cell wall and other structure will form in the megaspore. No, they'll form after some time. So that's why we are calling it 8 nucleate stage. Clear? Now this is what? This is 2, 4 and 8 nucleate state in state of embryo sac and a mature embryo sac. So think about it. This is the first mitotic division. So here 2 cells form. Now again 4 cells will form, and then ultimately 8 cells will form. Clear? Now these cells will arrange themselves in the embryo sac. They are not randomly present. So in the beginning they are randomly present, but after some time they will arrange themselves properly at a different end. So now look at this final structure. These 2 cells present at the center, 3 cells present below, and again 1, 2, 3. We can see 3 cells present at the top. So this part is which part of the ovule? This part is micropyle, and this bottom part is which one? Chalager end. Okay. Now how these cells arrange themselves, that we need to see. We can observe one more thing here. So during this cell division, there is a cell wall formation around all different type of cells except which is remain at the center. So there is no cell wall formation at the center. So 2 cells present as it is at the center, but in other 6 cells cell wall formation occur. Clear? So this stage is known as what? Any idea? First let's learn about this arrangement, and then we call this. Then we learn about how these cells and present themselves inside the structure. So think about it. First, look at the structure. Look at this mature embryo sac structure. We call it 7-celled 8-nucleate structure that we will cover. Now, what is egg apparatus? What is antipodal cell? And what is antipodals and what are central cell? See, 3 cells out of total 8 cells or 8 nucleated state, 3 cells present at micropylar end. Micropylar end, the end which is at the top. So 3 cells, let's say this is the micropylar end, okay, and at this end what will happen? 3 cells arrange themselves. These are what? These are integuments only. So 3 cells arrange themselves at micropylar end, one at the center and 2 around it. 2 around it. Clear? Now think about it. Another situation. And these arrangement of cell or collectively all these 3 cells are known as egg apparatus. We call it what? Egg apparatus. In this egg apparatus, one cell is egg cell. This cell is what? One cell is egg cell and another 2 cells are what? These are synergid cell. 2 cells are what? 2 cells are synergid cell. Let's revise it one more time. So out of these 8 nucleated cell, what happened? 3 cells will go towards which end? Micropylar end, which is present at the top. These 3 cells are collectively known as egg apparatus. Since it's an egg apparatus, so single egg cell is definitely present here. So if one single egg cell is present here, what's about other 2 cells? We call them synergid cells. These synergid cells contain some other structure as well, which helps in guiding pollen tube, the content of pollen tube towards the egg cell. Clear? Now think about it. And we call it filiform apparatus, that we will learn. Okay, it's written here. Synergid cell have a special filiform apparatus to guide the pollen tube toward egg cell and central part, right? Now let's talk about opposite end of what? Opposite end of ovule. So till now we learned about structure which is present at the micropylar end. Opposite end, we already know it is at a far place, chalagial end, right? So at chalagial end as well, 3 cells are present. Let's say this is chalagial end, and at chalagial end as well, 3 cells are present. And what we call it, since they are at opposite side, antipodal cell. So these 3 cells are antipodal cells. Okay, these 3 cells are what? They are antipodal cell. So out of 8, 6 cells are already covered. So how many are remaining? 2 cells. 2 cells. Okay. These 2 cells are present at the center. But I'll tell you one thing about these 2 cells. There is no cell wall division or cell wall formation between these 2 cells. So it is present as it is, or freely we can say. So these 2 nuclei polar cell is present as it is at the center. So what we call them? They are polar nuclei. Polar nuclei. So more than one nucleus in a cell. So we call it polar nuclei. Very important. So let's look at the structure or diagrammatic representation of mature embryo sac. Oh, it's a reverse structure. Okay, so micropylar end and chalagial end. So at micropylar end, 3 cells are present. At chalagial end, 3 cells are present. Okay. At micropylar, since there is an opening, from here the content of pollen tube will release. So here only we have what? We have egg cell, and with egg cell we have egg apparatus. So what is the difference between egg apparatus and egg cell? Egg apparatus contain more than 2 cells. So it contains 2 synergid plus 1 egg cell. So this is the content of egg apparatus. What's about antipodal? 3 cells are present at antipodal. And at center, what is present? Central cell, this is single cell with 2 polar nuclei. So there is no proper cell wall division between these two cells. So that's why we can say two nuclei cell is present. So we can observe one thing here. How many nucleus present? 8 nucleus, but number of cells are 7. Clear? So a mature embryo sac is 7 celled at nucleate. Clear? So that's all about the structure of ovule as well. What we have learned, what are the key points which we can understand from today's lecture? So first, what is the female reproductive part or the structure in the flower? It is gynoecium. Gynoecium contains what? Pistil. If there is one pistil, we call it what? If there is one pistil, means monocarpillary, yes or no? If more than one pistil, multicarpillary. If these pistils are fused, we call it syncarpus. If these pistils are free, we call it apocarpus. If we talk about structure of what? One pistil, it contains 3 parts: stigma, style and ovary. Clear? Now next is we need to learn what is present inside the ovary. Inside ovary, ovule is present. Now the part of ovule which connects to placenta, it is funicle, and funicle is again connect to ovule with a junction, we call it hilum. The covering of ovule is known as integument, outer integument, inner integument. The part which is responsible for nutrition is known as nucellus. The end or the opening where there is no integument, we call it micropyle, and the pole is known as micropyle. Opposite pole is known as chalazial pole. Right? Clear? Next we learn about the process of formation of megaspores from single megaspore mother cell, 4 megaspore produced by meiosis. Out of these 4 meiosis, 3 cells degenerates and only 1 cell remain functional. Now this 1 cell will again divide mitotically into how many cells? 4 cells and then 8 cells, right? And after this, these cells will arrange themselves. So 3 cells will arrange themselves and 2 opposite end, 3 at which end? Micropylar end, and 3 at chalazial end. The cells which present at the micropylar end is known as egg apparatus, and the cells which are present at chalazial end are known as antipodal. 2 cells or 2 nuclei remain as it is at the center, we call it central cell. In central cell, there are 2 nuclei. So total how many nucleus? 8 nucleus, but number of cells are 7. So we can say a mature embryo sac contain say 1 cell, or we can say it contain 8 nuclei, but the number of cells are 7. Clear? So that's all about the structure. But in the next class, we will learn about how the pollen grains travel from the anther to the stigma, and next we will learn about the process of fertilization. Clear? And ultimately the formation of seed. So that's all about today's class. Thank you so much, and in the next class we will continue the same topic. Thank you so much for joining.